Right, so is everyone happily fed and not so sleepy? It's a very difficult job to keep everyone awake, so I want to keep it as interactive as possible. Also, I want to hear people who have not said anything so far. So would be nice. This is your opportunity. We are not experts, it's just we have spent a lot of time in this field doing some of the stuff. So to propel the field in Indian context, I think it's very important. Everybody keeps their thinking hat on, asks questions, and there is nothing like a good or a bad question. All questions are relevant here. Right, so we have heard a lot about pathogen prevalence in the last 2 days. So I'm assuming everybody is quite comfortable with this parameter, prevalence, right? So it's just a representative proportion of infected individuals, whatever you sample in a population, right? So it gives you an idea what is there and infected. Prevalence could be with respect to anything, but we are thinking from the disease, whatever disease you are interested in here. Now, you heard a lot about community ecology, assemblages, how people are looking into detection probability and everything. For the reason we want to discuss this from the host context, I think something is very important because the infection dynamics for any disease or anything is actually driven by the host ecology. There are hundreds of parameters, hundreds of reasons, hundreds of factors which could be driving it, right? So can I ask all of you to, what do you think when I say host traits? What could be playing a role which could be affecting or driving this prevalence, say for a species or in a community or for an area? Just name those things. Can I have what people think about it? Anthropological factors. Like what? Like I go and cut the forest. Maybe one of the reasons. Come on, say from the host point of view. Any factors from the host? Huh? Okay,
host
so
immunity.
Somebody said immunity, someone saying food availability, okay, huh? Behavior of that species, okay, and physiological reasons, okay. Movement, very good, and sorry?
Structural variables, like physical structural variables.
Okay, so you mean from the biogeography point of view, the geographical barriers and from the host point of view. Okay, great. And
so it may be then the point of pathogen where it actually adapts and survives. There is a thing, the host must be susceptible to that pathogen, right?
So how will you define that?
So because sometimes the pathogen must see through the way where it can enter the host. So it can be any of the structural barriers things or any of the it may be any diseases things or immunosuppressant things like this. It may contribute to the prevalence of that pathogen.
Okay. What else? Come on, guys. There have been so many talks and everybody has kind of thrown lots of things into it. Population of the host. Okay, very good. So you mean population density point of view or whatever? Yeah? Huh? Climatic factors? Tolerance. Okay, very good. Right. Okay, great. So all are very good points. I mean, the way I have thought about touching upon all these things is in this framework. I am kind of trying to define it into ecological, biological, and anthropogenic, which all of you have put things into it. You all kind of understand what that might be doing it, but what the idea of this discussion, what we are having here is to understand or look at it more from the host point of view. So what I'm going to do is I'm going to take some examples for some of these things, and then we see how much can we think and how it can be applied in different situations. If you were to say design a study or do this work, and what prevalence would mean for that system, okay? So this is some kind of review. I'm sorry I didn't put the thing. So as you rightly said, the population density, population ecology, status, behavior, sociology, all of these factors can affect the parasite prevalence. And population size also refers to, it has to be of a certain threshold, right? for a disease to persist in a population? Would you agree with that? Why? Yeah, but say if you have an endangered species in some part of some forest, say Hulock gibbons, yesterday the talk was given, would you say that Hulock gibbons might be harboring, considering the population size of that species, same number or same kind of parasites as, say, rhesus macaque, which is very widely spread, or bonnet macaque in the context of southern India where we are right now? What do you say for that? What do you think about that? Huh?
Association may also.
We're talking population size right now, okay? For a species which is restricted to an area versus something which is very widely spread.
It can directly correlate because whichever species is widely spread, so climatic and all variable is also there. So parasite, pathogen also can be vary with all these things.
Yeah, but also there also can be a factor, right? If you have some very host-specific parasites which are kind of specific to those species, and if the population size becomes shrunk, you might lose those parasites also. So there are lots of actually theories, papers also people have done this work where they have showed that a lot of endangered species also carry very few and very rare parasites also. Yes.
Variability plays a role, genetic variability within a population. So the more variable are, the more things can go wrong or come.
Absolutely, I totally agree with you. And I think that's the second point I am also coming to here, is that when you have a very small population size, you are also kind of in a very weak population scenario, right? Could be because of your health, you're more vulnerable basically. You have a very small genetic pool. You can't fight with, you may be more susceptible. This is why all the endangered species which are confined to small areas, some islands or locations, they are very vulnerable. And there should be, say, some bird species living on an island, and it's a, say, water bird species. And if you have an avian flu, that can wipe out the whole population for that species. And another example, this is more from a captive bird point of view. Well, there's a whole vulture breeding program in India, or bustard breeding program. These are very highly endangered species. 99% population is gone, right? And they are doing captive breeding for these, the captive breeding program. They're spending millions and millions of dollars every year just to keep that population going and release them back. Imagine you have a disease outbreak and there is no surveillance or nothing going on. That can actually effectively wipe out all your captive population. So population size really, really matters, that how much can you sustain, how you persist. It can work both ways. You can harbor less parasite, but it also makes you very vulnerable to some new. But also there is a slow transmission because your population size itself is so few individuals is going on. Now, this particular third time, it has been talked a lot, right? Population density, how the increased contact rates increases, right? Somebody was also just now saying. And during Ashwin's talk also, we touched upon to the direct the transmission. But a lot of these things are kind of very bidirectional in terms of, as I'm saying, they're kind of interrelated, right? How population size affects the status, health status, I mean, and how it influences the disease, spread of the disease, and also efficiency of a disease transmission. Now this, you might say that why we are repeating some of the concepts here over and over again, but for the conceptually, for this understanding to go forward, I just wanted to put it out there that directly transmitted parasites, parasites which just rely on a contact with one individual to the other, they do rely on population density, right? Avian influenza is one of the examples. The higher the, and of course it depends on the different host species, and then frequency dependent transmission, which depends just on the number of contacts independent of the population. So there is no underlining factor with respect to the population density. Somebody said in the morning, which I heard, I didn't interrupt at the time, was that for vector-borne diseases, that mosquito could play a role. But imagine vector-borne disease is not a vector-borne disease, just a host and a vector. There's a huge element of parasite. You have to have a parasite in certain critical threshold in your population for that transmission to persist, right? Now that critical threshold or whatever to maintain, as I was saying other day also, that could be asymptomatic individuals and all. This is like kind of the holy grail. These are the questions that we are interested, trying to understand that what you are missing, what could be, if you have certain proportion of people infected, and what will happen if you have the biting rate of mosquitoes like that. So it is not so black and white as you would think that this will fall into this will fall into this scenario. But in the terms of disease modeling, if you're really trying to understand and nail it down, this is a fundamental thing to understand, that what kind of host ecology you have, what kind of mode of transmission are you really looking at here. It's just not that modeling and all will come much later, basically because a lot of these factors actually drive how you design your study, how you collect your data, and how you'll end up in analyzing it in the end. Yeah.
So does density dependent transmission or means have a high rate of infection, or frequency dependent transmission has high rate of
It can
infection?
go both ways. It all depends on the how the disease is spreading through a population.
Okay.
As I just said, if you have a lot of susceptible individuals for a particular thing, you will have a very high transmission rate. So this very dynamic thing.
Is there any disease which have both the density dependent and frequency dependent transmission?
No, malaria, I mean, a lot of vector-borne, sexually transmitted disease, which just relies on people. So there was a time when you have a lot of HIV infections happening in a population, right? Because there was not much awareness how it is being
Yeah,
caused.
but HIV is related to sexually transmitted STD. It don't contact with any airborne or anything like, right?
So Ebola is more of a fall into the directly transmitted, right? Because there is a lot of viral shedding going on there. If you have an individual or a host who have that virus, and if you come in contact with them, then it becomes, chances of infection are very high. So Ebola is more of a directly transmitted, which relies on the population density. That's why confining it is very important. Does that answer your question?
He's asking, is there anything which is both?
Not that I know of. No, not that I know of. Okay, so I just want to go back to what Varun was talking yesterday. If you really think about, he was really talking about that how you are going in the field and mapping the home range of different host animals, you know, elephants, tigers, all kinds of things, figuring out which is confined to which area. Can you guys just tell me that how that would be relevant from a disease point of view? What did you think of that particular aspect in terms of disease? Why is it so important for us to know? If, say, you are going to figure out the, say, disease of elephant or something.
Sometimes that animal itself maybe acts as a carrier or reservoir. It also carry parasite.
Yes. Anybody else?
If you study that kind of pattern or disease of which happen to elephants and any animals in the wildlife, we can predict the outcomes in the human surveillance, right?
Yes. So I'm just showing you this example here, but this can be applied to a lot of group living mammals or other species anywhere. This is a very long-term study done on badgers in the UK, right? Started all the way from 1970 till 2016. They knew a large part of this population. Why a badger? Does anybody know what's special about badgers? Do you know how badger looks like?
Oh, I just don't.
It's a small mammal. Badgers are really kind of one of those species in the UK which are not very liked because they carry bovine tuberculosis, right? So imagine you have badgers, and badgers, they're free living species. They go into agriculture areas, they interact with your livestock and cattle and everything. So imagine if you have these wild badgers going around, and if they're carrying these bovine tuberculosis, and they come in contact with your cattle and stuff, they can really transmit that disease. And livestock is a huge economic value, right, for any farmers to protect it. And there is a lot of, so one of the hypotheses what they had was that why so many cattle are having, and maybe if the badger population is so high density, maybe we should cull them. This is also a very big contentious issue, right? Why would you just cull something just like that? It's a wild species, it's just there, part of the habitat, and it mingles very well with the human habitation. So they went ahead, they actually did this mark recapture, something what Umesh was talking about with respect to his birds, that to understand the survival probability, you go to the same area, you catch those animals, you take samples, but also you are putting the GPS collars and stuff. Using that information, they have figured out these are the home ranges of those badgers. Now, badger is a very communal species. They live in groups. They have a group of like a male badger with the female and then their cubs, then other ones. So all these spots that you are looking, these are a individual family, basically. They are living there. Now, the question I want to ask you, which somebody was asking me yesterday, should you go and basically sample all the animals? Like, I mean, I'm digressing a little bit from the main thing what I'm asking here. Considering you have tuberculosis as a disease, but it could be any contagious disease. Just keep that in mind. I'm using the word contagious disease here, and you say sample maybe all the individuals if you have resources. Catching animals is also, they're doing it, right? So will that be a more feasible thing or just doing something? How would you go about it?
Feasible, because it can spread disease also while capturing them and
yeah, but you take
collecting
all
data.
precautions. People do SARS-CoV-2 patient swabs. The chances of infection is there also. Yeah, but these are home ranges. You know how the home ranges work? They have kind of a territoriality. They stay in that area. Yeah.
So maybe given that there are three regions here, four north and south, if you cover all three regions, like few bunches, like few sample sets, three families.
So each star is a family living there.
Yeah, so from each set, for example, core, you collect like 5 families, 5 from north and 5 from south. I think it will give you a good range of what is happening.
Right, but I would argue that's why would I invest in the whole family of the same place? It's a contagious disease. Huh?
Yeah, I think we can sample individuals from each of the like whatever the patches, and then since you mentioned it is contagious, I think doing individual sampling from each of the home range
Yeah.
would be sufficient
Yeah,
to.
I agree. So I'm just throwing it here. That's not what they did. They actually sampled most of the individuals, but I'm pointing it, the fact I'm highlighting it here that when you're designing studies like that, it really, you're up against resources, time, and everything. Somebody was saying that you go to a village, they just do the sampling from 1, maybe or 2 houses or something. How do you get around to that problem? Working in human habitation is slightly easier than in the wild forest, but yes, you have to find a way. And again, I underline the word, it's a contagious disease. Assumption is that the whole one family has other, maybe also having the infection, and usually that's what they found. So every time, if you really need to see, is the persistence of the disease is there, is the disease is growing, or, because remember, in this study, they just do not have prevalence data, they also have incidence data. Do you understand the difference between the two, how that works? Somebody from epidemiology background yesterday used the word incidence. I was very happy with that. That person is not here today. Does anybody, do you understand? What is incidence versus prevalence, and why a lot of studies only are looking at prevalence in the wild populations, not the incidence?
Incidence is the occurrence of the disease first.
Yes, but then, so.
Entry of the pathogen to the
So incidence is, again, since it's new cases, you need to follow up, individuals over a long period of time. So again, that resource constriction
Yeah, how much
is there.
new you are adding every time to a population, right? So which in a wild situation, if you're catching just the birds, say for malaria or for something, at the first instance, you will not have that idea unless you keep monitoring that population for a certain period of time. You should know the survival probability of those individuals. You have certain number of individuals marked, which exactly what they have. Say they knew that, I don't know what interventions have they taken, but what are the new cases which are happening, and are there new strains and everything. Anyhow, so this work was done more than 30 years now. This is like the longest long-term study done, where just to figure out, do we need to cull those animals and how do we protect our livestock? And what they found, imagine, we have talked so much about this population density as if one of the fixed effect here. Imagine if they were just only looking at the population and only some of these competition and things like that, they probably would not have figured out that actually it was the group behavior which is driving the infection dynamics for this particular disease in this particular area, in this woodland where they are doing this work. So what the point here is that if you were to handle this kind of a scenario, they probably will go by the either vaccinating if they can, some of the same individuals from the same family, or starting to cull from a point of view where you are taking a more strategic approach to do that, because you know that you can randomly, you can remove those kind of individuals from that population. Now, this is with respect to the badgers, but there are other studies which have been done in the wild animals, and what other factors can affect the disease prevalence, say within the wild animals, which could be with respect to their trait? Tell me. I'm giving you all the clues. Sorry?
Socially living animals, like for example, lions and deer and all come as an example. When you have social structures, you might have more.
That's a very good point. Yeah. What else?
I think there is a concept like some individuals are super spreaders. So they are in social living animals. There are some very like some animals are which are very social and they interact with other members of the population in that way. Yeah, group living. And then there is other like concept of modularity. I am talking from primate society. So modularity,
No, I agree.
centrality. So all these factors can.
Yeah, so in this study, if you read this paper, they have found there are some of the groups which were the super spreader versus because the disease prevalence changes, right, within a landscape. It may not be this uniform thing. So that's a good point. But actually, what I'm more asking here, more from the point of view of the how species are interacting in a landscape, say predators and prey, the kind of habitat they are using. So sociality is one of the factors, but here in case of this particular disease, which I have mentioned here, this canine distemper virus, it's a very serious disease. You know, once you have outbreaks, it can kill lots of animals. And the way it spreads that you have certain, it can be subclinical cases also in a population. A lot of lions are very susceptible to that. In India also, we have had some outbreaks, lots of lions died. But also in Africa. Now, when you are having a canine distemper, so canine distemper virus, as you can say, canine is for more to do with the canids. Lions are, imagine if you have some very infectious predators or other animals, could be hyenas or something, they are coming and they're feeding on this prey, a dead animal, but they are carrying the infection. But when you have another prey, other species, which is not the top predator, but a second level, like hyenas are more of a scavengers, right? When they come and they feed on the same prey, they also get the infection. So that's kind of a way of spreading, say, some same subtypes or something, not just within your, but also across in a different study. Vultures don't carry disease. Yeah, but not that we have know of. I mean, vultures usually died of diclofenac, not because of the disease.
Not all the diseases will get infected by consuming, right?
No, but I'm talking about really highly infectious disease, this canine distemper virus. I'm really more interested in the mode of transmission of a disease like that, how that is actually transmitting from a predator to a different predator or scavenger which is there in the landscape, utilizing the same resources, you know.
Does it a vector-borne disease or anything? What is the transmission like?
No. Canine distemper is only through individuals when they get through other infected individuals, and once you have it, depending on your immune status, like how healthy or whatever susceptible you are, some carry subclinical, as I said, no sign of infection, but others, when they get really seriously, they shed the virus through their fecal and other route, and once other individuals come in contact or when they are feeding on that, prey, they also get infected, basically.
A good example, because I don't know, but because the stomach acidity won't allow that viruses to replicate, right, or to spread it, right?
It might be true for some of the viruses, but in the scenario I am talking here is something which spreads within the wild species.
Yeah, because every virus has a route of entry. So,
yeah, but see, lots of viruses, either they get transmitted either through vectors, the mosquitoes and all, or through the direct contact or respiratory or other factors, right? So Here it is more through the shedding from the bodily fluids or other individuals coming in contact with that. Yeah? No. That was more of a, yeah, tumorous thing.
No, not basically they fight a lot. They scratch.
Any question? No? Yes.
I'm just wondering, just like since you're talking about two different predators, but also, I mean, I was just seeing a photo yesterday where someone had written about how langurs eat leaves and drop it down, and which cheetahs also feed on.
Sure.
So that can be another means of transfer.
Why langur and cheetal only? When you have bats feeding and you have a half-eaten fruit go down, then you have pigs there or other humans, they
Nipah,
don't
yeah.
wash, then it's a very direct transmission. Don't go to langurs and cheetals. Nipah virus transmission has lots of reason or other viruses going around in that landscape, right? Okay, so there's a very interesting thing which I want to talk about here, what's happening here in this 4 figures here. So what this study really tried to test, or they showed that so you know, when these animals, when they live in these social groups, not all the groups in a wild population are of the same level. Do you know that? There are some are very dominant groups, some have a very high status, and some are not in the same domain. So what does that mean when you have a high status? You have better access to resources. You get to kill first. You basically call the shots in that landscape, and the others were just, I mean, have you seen Lion King, how things they have shown in that? So what this study was showed that all the animals which were in the cubs, which were born from the females, which will belong to the higher status, they had very low infection probability as opposed to the ones which were in the low. And that makes sense because you are not in the high status, you do not nutritionally so well off, you are actually contracting that infection very higher, this thing. But at the same time, the one point they also made there that high status individuals, especially the males, because they are defending their territories, they have to be in contact with so many different individuals. They are also very high prone to the infection, basically, because they need to manage their land there. And what also they showed that the same thing applies in the infection probability for the cub for here with the pre-breeders. But also it affects their survival probability also, that high infection group, high status group has a very high survival probability versus which is the cubs in the low. But look at the survival probability which comes here in the cub. Again, it's kind of diminishes in this. So the point over here is that host ecology, the way you live, the way you are using your resources, is really drives what you can get infected with and what chances you will have for your survival in that landscape. I mean, if you put it into the context of human beings, I mean, a lot of people, if you're not nutritionally fed, if you're very stressed out, you get sick very fast, basically. It really suppresses your immune system. So having that balanced way of life is very important, but this is all wild world. This is how things are going here, yes.
So that incident rate will change if this behavior...
This is the infection probability, prevalence, yeah. They are not looking at the incidence here. This is the only prevalence, presence and absence of the virus, what they are looking at here, yeah.
I just want to know what pre-breeder means.
Pre-breeders are the ones which are not sub-adult, they haven't really started breeding. They're looking at the social structure, but also individuals of different age in the population, how they perform, because that really guides their chances of being in the next, set of generation, what will they guide it. Now, I can't get away without showing these slides since somebody talked about the avian influenza, and this is something I just want to, I'm throwing it in here just for the sake, because you asked about avian influenza to Umesh, that why and how, why is he planning to study and all of that. This is something I did long time ago, but Avian influenza, I mean, as you all know, that it's very, very prevalent right now. A lot of UK seabird populations have wiped out. It's doing a havoc in America. I cannot believe that it's not there in other parts of the world, but I think places where you have very good surveillance, it gets reported, and good money is being thrown into figuring out what the genome looks like, what species are infected and or whatnot. With respect to avian influenza, Mongolia is very, very special. Mongolia doesn't have much poultry, actually none at all, really. You can actually draw the boundary of this country by plotting the poultry distribution. So, because the reason for that, Mongolia is more of a nomadic population. You know, they live in these kind of gear camp. It's a fantastic place to work. I would love to go there again if I can. And it's kind of a very iconic place. And I would say a perfect natural laboratory if you want to understand that, is it really the wild birds which are driving the avian influenza infection? Because every time, whenever you have an outbreak, it's usually the wild birds which are implicated for the outbreak, right? That, oh, the migratory season has come and the birds have brought the virus, and then that's how we are going to have the infections. So we did this survey. So there are lots of natural salt lakes in all over Mongolia, which are very nice. They get frozen in the winters, but when the summer comes, then you have a lot of birds which come and they breed there. Birds like gulls, most of the water birds basically. There are no trees there. Remember, it's all water birds. And usually the way the migration here works, these birds are coming here from the China side. So if you have an outbreak of avian influenza, say in one of the poultry markets in China, you can expect some of those birds coming here with the virus, and you might see some of the dead birds in this landscape here. So one of the questions that we were trying to answer was to understand what kind of prevalence are you seeing in what species? Is it really the migrants ones which are breeding? Because most of them are migrant, and how does that correlate with the say the outbreaks in China? What kind of seropositivity or serology? Just the normal questions which help you to understand how this might be driving. So we surveyed these landscape. We didn't catch any wild bird per se, but what we were doing, we were looking at the fecal samples around all the lakes. So you go around the lake and you collect the fecal sample, and you can do the whatever you want, basically PCRs and whatever. And then you also see these dead birds, like this one of the swans is lying there. And there were very trained veterinarians also were there on the team, and they were recording, and they were also doing necropsy, because most of the birds which were died were actually of H5N1 positive. So they are wearing the whole full body suits on that. What we found, actually, this was very interesting. This work was done over a period of time, good 5-6 years, is that, of course, that year that you had outbreaks in China, that did coincide, but some of the times you didn't have the outbreaks, yet you found some of the birds when they come and they start breeding in those areas. Can you tell why would that be happening? How those birds might be having the infection? How can you have an outbreak when nowhere else are you noticing anywhere? There are no poultry, right? You can't blame them also.
Maybe because when the climate changes, suddenly you would see the outbreak, and when the climate again changes, you won't see it.
What does that mean?
Some seasons are more susceptible. The organisms become more susceptible in some seasons than others.
1 year of climate change, like last year was fine, and this year you have.
And it's very seasonal, the migration and all is very
Yeah, it's very seasonal. They come only in the summertime to breed, and then they go.
the same time.
Can you repeat it once?
So I'm saying that 1 year you have an outbreak of influenza, and it kind of correlated with, because you have outbreaks happening in China. Next year, we didn't hear any outbreak in any of the main area around it, but yet you notice there was an outbreak happening when the birds came, and you have these mortality going on. Because by the time birds will come, they start nesting, and they all are very group, congregate and thing, and by the time they're breeding, you have sub-adults, and you see like suddenly the birds are starting to die, and you see these swans and things lying around, including some shovelers and things.
Previously infected area, if they're coming in contact with that, the virus is circulating in that area.
Does it have to do with the migratory routes of different birds? Because maybe a lot of different birds are, maybe a lot of different migratory routes are going above Mongolia, and multiple birds are coming from different locations together.
True, I mean, it's possible. I mean, I'm not ruling it out, but a lot of things, what we found that because these lakes, these are the salt lake, they get frozen in the winter. They go really minus 40, 60. So that time when you have a virus particles, they get frozen. Yeah, it's a natural freezer. So when next year they thaws, it's quick enough to infect whatever is coming in those populations. And this what they described was after a good few years of study of the work what they did. Now, going back to your flyway question, this is how the flyway looks like. Of course, we are here in the part of the Central Asian flyways, and India attracts so many different species, and somehow the flyways drives, some of the patterns you see in avian influenza. You can say that if you have lots of outbreaks happening, and if you are going to have birds from that part, then you can expect. This is one of the species of bar-headed goose, which is, it flies really high up and it also breeds in Mongolia. We get pretty much all the global population in India for this particular species. This is a very lovely figure which Bird Count India have made. And it just shows you how the whole population, and they are very susceptible to avian influenza also. And the birds say they come from Mongolia, they stop here somewhere, then they kind of filter through all the thing. Now, this is a figure I made a couple of years ago when we had very mass scale outbreaks of avian influenza in our country. Can you tell me what is very interesting about this? Which somebody has said, I made this talk keeping a lot of things which came up in the last 2 days. So thinking people, if they have questions, we can answer. So what do you think, what is so interesting about this figure? What do you see, apart from the dates I have put on the map? True.
Most of them are water birds. China.
I'm trying hard to keep everyone awake and myself.
There is a different variants of H1, H5.
What variants?
There is one H5N8 variant. There is one thing, and H5.
I'm talking more from the host point of view.
Host point of view.
Remember, we are concentrating on the host right now. Huh? Yes. So what I found very interesting about this. These are all the cases which were reported everywhere. Every day you will have a report in newspaper or some thing which is saying that, oh, there is an outbreak here. So the first outbreak say somewhere started here in H5N1 here near Pong Dam in Himachal Pradesh. Then you have, these are all the poultry, the farms where that happened. What I found that, yes, it is right that most of them are water birds, but also what are these crows doing in here? There are a lot of scavengers also involved here, right? So the question when you asked from Umesh saying, are you looking for avian influenza? I think that's a long lost cause, because avian influenza, the probability of going in a eagle-less sanctuary in a very woodland is very, very remote, because it's a species which likes water, because that's how the transmission route happen. When you have a lot of birds, they come in, congregate, they're shedding the virus, other individuals are coming in contact with them, and that's how they're getting infected. Crows are getting infected because when they see a dead bird, they go and they feed on it. And that's the perfect transmission route. So again, I'm dwelling on the same point which I was showing for the hyena there with respect to canine distemper, that there are not just 1 or 2 modes of transmission, there could be N number of ways a parasite or a pathogen might be transmitting from one species to the other or in a population, basically.
Yes, that's also why ecology is so important.
Yes.
So for example, hopefully in this course, you do not think that you can look for any pathogen anywhere.
So while feeding, it may enter through the nasal cavity, right? There's a chances of entering through the nasal cavity.
Sorry, say again?
While feeding, it may enter to the nasal cavity because it may be the route of transmission.
Yeah. So you know what is happening in these areas? These are very highly, densely congregated areas where you have multiple species are kind of using. So say imagine you have a couple of these species arrived, and if they are carrying the virus and they are super spreader and they are going around, they're foragers, by the way. They don't feed in the water. They forage on the grassland all around the wetland. Well, this is why it's very important to understand your host species also. If you're looking for parasite, please do all your homework. It's just not about that pathogen at all. You need to know everything about it. The species, say barheads are going around, they're feeding on this grassland, wherever they're going, they're pooping. They have other species which are coming, they are using the same area. It's a perfect place for the species to get infected with those viral shedding going on in that area. Plus, you have these pools of water around where birds might be bathing, fluttering, whatever. We take samples, from these sites of the thing, and if you screen them, sometimes they are positive for different viruses. It's a very good sample. You don't even have to touch the birds. It's just there, basically. So that's the intensity of the infection goes on, yeah. Here, this one, I don't know, maybe they didn't report. One thing, have you seen what I'm writing here, potential? In the news, it was labeled as avian influenza. The reason I'm calling it a potential because they were not tested on time to say that these were all deaths from the avian influenza, but they were, because that's a long discussion some other time I will talk about, but this is how it is. So it is what is reported. I'm not saying they may not be. So don't take it by the face value, basically. Right, so the other factors then, I just want to say that we have had so many outbreaks in our country suddenly. 2006 was the first time an avian influenza outbreak was reported. Since then, you had some really very large and some very small. And one of the reasons that a lot of wetland habitat has been shrinking. It's been used for urbanization, pressure is being so high. People are using more poultry farms. They are more vicinity with the wildlife areas. And you have very high chicken density. There is no regulation also. So there are lots of factors, which drive. So if you were to design a study to understand avian influenza ecology in an area which areas could be more prone for an outbreak, I would suggest I'll start with something like that to understand where the previous outbreaks were happening and how that is with respect to some of these conditions, what might be driving those outbreaks. Beyond poultry setup, because poultry are the last point, because when the outbreak happen, they just go for culling for those birds. Now, I also talked about that a lot of time they always blame the wild birds for the outbreaks, right? Like in whether they're coming or not. This is some of the avian influenza is also a very notifiable disease. I mean, whenever you have an outbreaks in our country, they must notify, and there are very few labs who actually record those cases. And it's there on the World Organization for Animal Health. There's a big website. You can go on that portal. You can actually download the whole. Not FAO? No, it's under FAO only. You can download and you can say which areas have had these cases. And this is Walsh, I think David Walsh. They did this summary of figuring out the spatial risk, how temporally spatially it spreads. I find it very interesting because yes, you have outbreak in the high season, as they call, when the migrants are coming in the country and also when they have left. But look at this area, all the blue. This is May to October. You do not have any wild birds coming at that time of year. All of these outbreaks have happened in the poultry sector, which means, what does it tell you? It is an endemic. It has become endemic. There is a lot of circulation happening within the country now in the birds, which is leading to these mass mortality, and those must be culling those parts. So all these factors, they happen, and I think one has to look just outside the box to understand how some transmission might be happening and they be affecting. And it's a very high important disease in terms of, because the kind of poultry sector we had in our country from that point of view. Now, the second thing which I want to talk about is the migration and colonization, how that affects the parasite prevalence. Thoughts on that, anybody? How that might be happening? Somebody said something might be going somewhere, taking the parasite. Come on, Art Park. Come on. I just want to know your thoughts before I dump you with lots of information. Otherwise, I'll be singing the song here only. How migration might be doing this? We just talked about actually. Yeah.
Tree birds could be carrying vectors.
I told that already.
Oh, sorry.
Beyond that.
Yeah.
Very good. Yeah. Yeah. So migration is very expensive. I mean, I will talk about it in a second, the whole thing behind, but as a life history trait, you choose to migrate. Some species are going short distance, some are going very far. You're not just spreading your germs, you're also exposing yourself, right? When a species makes that journey on an annual basis, you don't know whether you will be coming back because you're going for these new sites where you are kind of evolved to use. And then, so there are a lot of studies actually which people have looked into this red deer population. This study is done in Europe, which actually looks into the tick load, the ticks which deers carry, how this migration, the seasonal migration kind of affects how they carry different ticks. I mean, ticks are very much, responsible for a lot of tick-borne disease, Lyme disease and things like that. And I think that was the main reason why this study was done. And as you can see that the animals which are going farther away, they are having very less tick load as opposed to which decides to stay where they are. So if you just stay where you were, your tick load is very high. But also that also translates to the elevation point of view as well, which I thought was very interesting because it is one species, but some individuals are deciding to move away and some are actually there in that same area. And that's why it makes it very interesting to test this thing, that how the tick load actually changes. But I also would encourage you all to read about this. There's an animal migration and infectious disease risk. It's a very lovely review done by Sonia Altizer, where they talked about how the different species, how many miles and stuff they are going, and what kind of different parasites and threats they are actually faced. So for example, look at this gray whale. It's an 18,000 kilometer journey, the migration this one takes. And it has a lot of endoparasite worms and everything. Yeah, it's a very nice review, and same for the butterflies, which she actually specializes and work on that, how these migration, the butterflies, they carry different parasites. You can go back and read about it. I will not dwell too much on this, but I want to ask you one very fundamental question. Is that why do you think animals migrate? Huh? Why would they force to migrate? Not everything is forced to migrate. Crows are here. Habitat is disturbed. Okay.
When there is a harsh climate, in order to escape from that harsh climate and food availability, so they will migrate to a particular, and breeding also, mainly for breeding.
Breeding and food is related, right? You have to have food to breed. Oh, yeah, I guess. What else? Yes?
Tracking resources, but also since you mentioned how the ectoparasites or ticks are changing in individuals, I think that's also an angle. I mean, I am thinking about it for the first time now, that maybe it's because also to move away from diseases, but also you're saying some individuals are staying back.
In that particular species, they do.
Yeah, so that goes true for a lot of other migrating species also, like even I think black kites for that matter, where some individuals do stay as residents. But could it also be because these species are gotten more immune to the diseases that spread in the...
Yeah, but that example was more to do with just the tick load, right? Not an endoparasite as such. So the fact that you decided to stay in an environment, but you're exposed to so much of ticks, it just shows that it's a decision which that particular population has made and they are not moving. So it's right that a lot of animals, they move because of intolerance to cold temperatures. So they go from very cold environment to the warm environment, or warm to cold reason to exploit food resources. But I'm not interested in all of that. I think one of the things which has been really debated and also shown in lots of species is that some of the species, or some of them evolved, actually the species we have been studying is to escape the parasites, basically. And I want to talk you through, on this particular study, it is a very beautiful study. I would encourage you all to go back and read. So this study is done by a group in Lund University. And what they showed here is that you have parasite richness, which a lot of people talked about the patterns, right? So there's a richness is very high here in this part of the latitude, but as you go to the north, the parasite richness also goes down. But that also translates to the host species, the richness, what you see in the host. What they have done in this study, they are looking into the colonization and the migration pattern of the species. Do you know how the colonization has evolved or worked, basically? Why some species are there and they have colonized, right? And some, they colonize and they establish. Those populations never migrate. But then you will have a sister species of the same group might be making that long journey over and over again. This is from an evolutionary point of view is a very interesting thing which lots of scientists are trying to understand, that why some of the populations migrate, say, only to certain area, what's there with their biological clock or whatever clock, and versus why they are decided some of the thing. So in this study, what they did, they screened lots of species, but I'm just highlighting these three. These are 3 species of pipits. These are small species. The one of the species which is confined to the Palearctic region, which never migrates. The one in the middle, it is in Palearctic, but it migrates. But then you have one in the African sub-Saharan region. So you know that in lots of species from the Palearctic region, they do migrate. They go to the sub-Saharan Africa for winter, basically, for the same reason that it gets too cold and they exploit these areas. But not all. There are a lot of resident species which don't. So what this group did, they looked into the pathogen diversity, as a function of this latitude, what they have across different species. And what they found that when you looked into the immune genes, what they have, the MHC1 class gene, what we were talking about in the morning, is very diverse in the African birds, the ones which are making this long journey. Because you have a very diverse set of genes because you're exposed to lots of parasites, so you need to have them to protect yourself, as opposed to the ones which are migrating or the ones which are only in the Palearctic region. I thought this was a very interesting way of showing that how some of the species, they decided to migrate and then evolved mechanism to deal with the disease factor. Right. So now, any other question before I keep moving on? Is that fine? Okay, yeah. You need mic. I can't hear you.
Sorry, does that mean that migration is actually allowing them to evade some diseases?
Yeah, but it works both ways. You are evading that, but at the same time, so imagine if a sub-Saharan, the bird which breeds in sub-Saharan Africa in the summer, but it is moving to the cold region in the winter. So it's too cold there, so maybe very less parasites there at that time of year. But when it comes back, it needs to deal with whatever is there, basically. And then at that time, you have everything coming from everywhere. So you are basically exposing also yourself. But it's a kind of trade-off, right? Probably the food, the habitat thereafter is so rich that they have to chase that. They are kind of ignoring the parasite part, and some have decided not to in some ways, basically. So other thing which actually affects the prevalence is also the competition and the predation, as I just mentioned, and all of these factors, they kind of play a role in that. So this particular study, which I have mentioned here, is that our predators is good for the health, for the ecosystem. And this actually goes back to the rodent study, which we've talked about. We're looking at the Bartonella and different gradients of habitat. People are interested in viruses in both rodents, but also, say, in bats and everything. What this particular study was, it was showing that predators, when they feed on some of the rodents population, which are supposedly in very high in the risk factor for the different diseases, they're actually doing a very good job in reducing the risk for some of the species. And the same point actually was made from the host traits, how in different habitats with respect to these shrews and bats and the other rodents. This is also very good study. They said how rodents actually harbor much more viruses, the shrews, than what you have. So imagine if you have these different high diversity of shrews and viruses when you are living in your, say, rural or kind of areas, and the potential risk can be very high. But at the same time, a lot of area is getting urbanized. This whole land where we are sitting here, it was a agricultural land, and I won't be surprised that there was some kind of wildlife was here, and which has been kind of evaded, and we disturbed that landscape, and you still keep coming in contact with some of those species and exposing yourself. And I think that's the very fundamental thing from the point of view of urban ecology as well, when people are trying to understand that by disturbing what thing where, you might be increasing the risk for what. This is why you need to have a very detailed understanding of your habitat and the system as well. Now, with respect to the competition and also predation, I thought this was a very good study, which they actually looked into the density here, density of some tadpoles, but also the presence of a predator and a nematode infection, which actually it's a three things here going on. You keep increasing your density in a experimental setup, and how that might be driving some of the, can you tell what might be going on here? How having lots of individuals of your kind in a bowl can affect your development rate, or having competition with the others can affect your proportion, how. So all the points here are no predators. This is with the predators. So when you have high density with the predator, you have time to metamorphosis is less, but where there is no predator, it's very high. No, not impressed with this answer? Sorry? What is what?
The black and white
exposed
dots.
ones are the solid circles, unexposed are the open circles. That's written here. This is just a study which I think is very hard to do in a very wild situation, to understand that if you have a situation where you have a high density of host density, but also infection going on, and predation also at the same time, what might be driving what in a situation? And something like that can only be tested in an experimental setup where you can say that how far you will increase. Yes. Say again?
They are in tadpole stage, the immune system is very weak on that time. After they become adults, the immune system, they put the energy into development of the immune system. Maybe that is the one factor.
Yeah, but I think here what they are trying to really say that how you speed up your processes in order to develop faster so that you can survive really, if you have that kind of situation going on, that you are competing with the individuals of your own kind, but also you have a predator which is sitting there, but also there's some infection which could drive the population down. If they have some innate immunity or something going on, that might protect them. But imagine this is just a very lab population where they are trying to think something. You have a question? No, okay.
Hello. Are younger these tadpoles taken care by their parents?
No. This would be just
They're
thrown in
independent.
of a certain stage. That's it. No, there won't be any parental care involved here. This is just like you take 15 tadpole here, 30 tadpoles here. Then you introduce them, expose them to different pressure group, which is the predation or the infection with the thing like that. Yeah. Which I think Kanika, you guys also do, no? Something like that. Can you explain some of that? Mike.
Yeah, so we also do similar work. I mean, we don't expose them to parasite. We just
Yeah.
have predators.
Hmm.
So
So you need to tell your host species.
Oh, right.
Yeah.
So the prey is Aedes aegypti larvae and the pupal stage. So the immature stages of Aedes aegypti, they are the prey, and predator is dragonfly nymph. So we actually, that's what I was looking at. We have also observed that under predation threat, uh, so it's so the predator is not free, it's just the predation cue that we provide. So under predation threat, larvae develop faster because the dragonfly nymph is gape. They want to develop fast so that they don't get eaten up by the predator. So that's what we are seeing.
So you also have in the high density the.
No, we just do it at one density because we are also looking because we are looking at multi-stage trade-off. So it's not just larva adult, it's larva, pupa, adult, and different combinations.
I see. Okay.
Yeah. So
also, you have three different stages.
yeah,
Okay.
so we are looking at trade-off at multiple stages.
And which one works best?
So what we have found, I mean, I'm still writing that one.
Yeah, you know the results though.
So, but it looks like the mosquito larval stage is, it's also very intuitive. It's well equipped to manage trade-off. So when it gets exposed to a predator risk, it's able to develop fast, and once it becomes adult, females are doing better than males. Like
I see.
the
So there's game.
adult lifespan of female is almost as same as the control females.
Right.
Males also develop fast, but then they die all faster.
Right.
So for them, the trade-off is worse.
Yeah.
Also, if just the pupal stage experiences threat without the larval stage, the trade-off is worse. It's not, alone, it cannot manage the
Right.
trade-off. It's good that it has that experience from the larval stage.
Right.
So pupal stage alone cannot manage trade-off well. With the
So
larval
they
stage,
have to have that experience in order
yeah,
to.
yeah. And it makes
Very
sense because
interesting.
it's a shorter duration stage, it cannot feed, so it
Wow.
becomes difficult, yeah. But we
They
don't
have,
expose
they have
them.
good memory.
Yeah. We have also seen that the pupal stage has memory. That means if the larval stage has already experienced predation risk, before they pupate, if we take them out of the container where there is risk, pupae still respond to that risk.
Right.
So there is some memory which can pass over.
Yeah.
Yeah.
Great. Okay, so if nobody has question, can I just move on? Yes. Yeah.
If you insert one predator, so that predator will eat all 15 in 1 day or 2 days.
Well, it depends. You should read the paper. I think they control for it, how many they introduce at what time.
Because how they will control that thing? That's
No,
why.
of course, I'm sure they have taken care of that. Okay, so it's 3 o'clock. I think I do not want to keep going for too long. I really want you guys to have half an hour break. Okay, so I have a few more things to cover, and we'll go. And some of the things I'll skip because Ashwin has mentioned. So again, in terms of the prevalence, when we are talking climate season, everybody has talked and understood from that point of view. So long dry season and low temperature, that kind of affects the limit of survival of parasite and development, which we understood this morning. Also, climate itself affects the vector, and all the host and other abiotic elements part of that. And as rightly Ashwin also mentioned this morning, like that high temperature can increase the parasite growth, right? But it is not a linear relationship. After a certain point, that it will drop down, and it will also increase the parasite mortality. So climate change will work for some of the species, and some of that may not thrive. So you should expect to change in the distribution of the parasite and vector species based on this kind of understanding if you have a very fine scale data for this environment. I'm not going to go into the detail, but I just wanted to put this point which was asked, that how the parasite are fixed on different vector species. So we were talking about that how this divergence has happened for Anopheles mosquito, for falciparum, and then you have this whole bird group has evolved and they all are fixed on different vector species here. Right, so let's skip through that. Now, I just want to mention a couple of things that we very nicely, Ashwin talked about this whole Hawaiian system, right? That you have these species which were introduced, and there was an element of low immunity in those native birds there, and that led to the extinction of so many species, and some are still struggling to survive, right? I just want to give some example for some of the work we have done here in India, which was part of some of the studies which I was doing 10 years around. It all kind of, in addition to that work, I did my postdoc on Hawaiian stuff long ago, and that really made me think that if you have that scenario happening in the Pacific Islands for those birds, will there be environments or locations in the world where you can expect that to happen? So for example, Himalayan birds, you have such low temperatures there. Could you have species which may have very low immunity, and probably they are living without any of these blood-borne parasites, because malaria is so prevalent everywhere. And because of the cold temperature, you should not expect many mosquito species to thrive after certain temperature, right? So this was like the basic thing I had in my mind, and we worked towards it. And there are so many European birds which come and winter in, they don't go to sub-Saharan Africa, by the way, they come to all Indian subcontinent. And a couple of them are very sympatric, as you said, they share the same kind of habitat in terms of distribution, and they also share the parasites. This is also some species which common rose finch come, and then when it utilizes the habitat, it mingles with some of the Himalayan rose finches which are found in the high elevation thing. You guys might think there are a lot of bird talks, but we are bird people primarily who do a lot of this work we have done. So this is a thing which is, so a lot of species which are kind of, we were looking into from say 500 meters to 3000 meters of elevation, and you have all these different kind of species. This shows you the resident versus the elevational migrant. So this is the distribution of species in the summer range, but in the winter range, and in the summer it moves down. Remember how Umesh was telling that some of the species, they move down during the in the winter time because they can't tolerate the cold season. But so the same thing happens in these birds as well. Now I just want to say that, so this was the whole system we derived to understand how the things work. It all involves, we are looking at the parasite intensity and everything, not just looking at the PCRs and everything, but also looking into the hemoglobin, which I will tell you why it was important with respect to these birds. What does hemoglobin do? If you have malaria, what does hemoglobin do to you? Decrease? What does malaria do to a person? Does it affect your hemoglobin? Yeah, so imagine if you have infection, what will happen? Hemozoin, yeah, and parasites like to exploit that basically. So while catching these birds, looking for the parasites, we were recording different parameters by doing all the morphometrics and all kinds of things. We've done that. This work happened for over 5 years. We caught something like 5,000 birds. Also looking for different vector species. This one point I just want to mention here, which was asked in the morning with respect to the malaria, why you do this work when, basically it's very important. So if you want to do, so pretty much all the studies on malaria which happens, if you're doing a very seasonal work, happens during the breeding time. Because that's the time your immune system is so suppressed that parasitemia will be very visible in your blood. Do you understand how that works? Your immune system is very compromised, you're very stressed out. So if you are infected with those parasites, if you sample these birds, it will be showing up in your blood. As opposed to in the non-breeding time, when you are not kind of stressed because there is no pressure, unless you are going through a very full-blown malaria, it will be fine. This is why a lot of this work we do was in breeding time, just to make sure if you're finding, the chances of probability of finding the infected birds or seeing that birds, say high elevation bird, if you say they have no malaria, because one of the hypotheses that we are testing, we should be able to prove that. So this is in the 500 elevation. Somebody asked me that is there a temporal variation? Yes, it does show a very good temporal variation across the season. So in the spring versus in the winter also, but not so much for the, it's very not so pronounced, but it is still there. But it is there. So the one thing is that it is there throughout the year. It's just sort of, it kind of changes. we had very low prevalence, but what you see is a very negative elevational gradient with respect to Plasmodium, because there are no mosquitoes in the high elevations. You should not expect any Plasmodium to be there in the high elevation. Same for the Hemoproteus. It is transmitted by these black flies. It doesn't. But look what it does to some of the resident birds for the leukocytosome. So leukocytosome is transmitted by these black flies, and they like breeding in these very cold streams in the water in the high elevation. That's why most of the resident birds are infected with that. So this is the part of the kind of a jigsaw puzzle we were trying to bridge, that where is the parasite, what's happening with it, and how does that relate to different mosquito or different vector species, what you find in this landscape, basically. Now coming back to this hypoxia thing, which I just want to spend 2 minutes on that for you guys to understand, that a lot of high elevation birds, they can regulate their oxygen intake. So when they are going into high elevation environment, they can actually increase their oxygen. When they come to the lower elevation, they have the ability to change that. But what happens for a species if it gets, say, infected with the malarial parasites, it can really compromise their ability to do that. And that is something we were trying to understand, that does that put them into another kind of a pressure or a risk, that you have to physiologically deal with this change, which you are evolved to do that, but parasites were not there, right, when we in that phase. So we did this work and we found that resident birds kind of do not have that much of significant change, but the elevational migrants, as you expect, because they are going up in the higher elevation, they do change and they regulate that. But with respect to the parasites, we found that only the birds which have these mixed infection, a lot of species get infected with more than one different kind of parasites. You see them all the time. So it's not just one Plasmodium or Influenza, but it's like a mixed infection that could be more lethal for you to regulate your hemoglobin, the content in these populations. We did this. For this, I'm going to skip this, which he was saying. And also in terms of the host specificity, that a lot of species, they carry the parasites of their own kind, which are very phylogenetically closely related to each other. So this is something not very new in the context of that, but it just bridges the idea that birds, they do show similarities if you are a very host specific. How a lot of cooperative breeders, for example, they're living in the same space, they carry the same kind of parasites. But also you see some kind of spillovers as well, which are not found in in different species as well. This I'm going to skip. Now, this was one of the studies which somebody talked about here, is with respect to the food availability and the diet impact on the prevalence of this particular parasite, which is Trichinella britovi, which wolves, this study was done actually in the wolves to understand that wolves, when they are coming in contact with your livestock or other animals, how are they putting others at the risk, or which part of the forest they might be more prone to having this infection. So because wolves are being culled as part of this work, they have been screening the tissues or whatever they were getting as part from all the people who were doing the work. And when they screened those, they found a lot of these parasites which the wolves are carrying actually is part of the sylvatic cycle. Do you understand what is sylvatic cycle when I say that? So sylvatic cycle for a lot of parasites is something which is restricted to only in the forest species, but not part of in the main urban areas, or say in the villages in this context. So I think a very good example for that, I would say, is dengue, how dengue has evolved. Dengue actually used to be only a sylvatic disease, which was evolved in the primates in the Africa, but also with a very specific mosquito species, which is found there. But the fact that humans started going and all the habitat, change happened, bushmeat, but also changing the landscape and everything, which allowed the parasite to jump onto the humans. But at the same time, a mosquito species became more adapted to the human habitat, and it became more adapted, and this whole transmission for Zika, chikungunya, and dengue. And that's how the sylvatic cycle is still there. The wasp you have is more of an urban cycle, and that's how it has become so kind of fixed in the human population now. So this kind of studies, when people are looking into it may be lethal, it may not be lethal, but they actually helps you to understand where are the risk areas. Remember, we were talking and thinking about it that you don't have to go to a cave to understand the risk for a disease. Some of the things might be happening at your doorstep, how people are changing the habitat, but understanding who's carrying what and defining or what could be the drivers, that's what the ecology really allows you to do, I think is the holy grail here. I just wanted to mention this particular work, which Umesh didn't mention yesterday. So this was based on the Eagle Nest Wildlife Sanctuary. When this work, we did it in collaboration with Umesh, basically here. So what we were looking at that, if you remember that he was talking about these plots which he had for logged forest versus unlogged forest and looking into the survival probability for the species and birds. When we collaborated with him, what we wanted to understand, so these are all 2000 mid-domain kind of location where you expect everything to be very rich in kind of species diversity. We screened those birds for these different malarial parasites, basically to understand what might be driving the prevalence of these malaria in these different kinds. And this is just for you guys to understand, since we talked about so much of modeling here. This is the kind of model we kind of built around it, that looking at the log status for that particular grid where you are doing the work, which is one of the fixed effect, the feeding stratum where the birds feed, how far they go and up. So we did run a number of models, and actually the top model which shows that it's the social behavior and the foraging structure which actually kind of helps you to explain that leukocytozoan infections are much higher in, say, for example, in canopy dwelling species. And that makes sense because a lot of the vectors which the leukocytosis is transmitted, they are very high up in the canopy as opposed to the plasmodium, which is more in the lower stratum. And I think this is something which Ashwin also talked about. And same kind of thing reflects in your social structure, whether how solitary and how group moving species. Do you move in a mixed species flocks versus, because that's where you are kind of exposing yourself and coming in contact with same kind of vector species. I just want to talk about this one thing for the non-native species and invasive species parasites. So there are lots of birds. This is not migration, this is not colonization. This is people actually going and introducing species into new places, basically. And Hawaii, actually, we talked about that brought so much in introduced species. Common manna is one of those species. In the morning, house sparrow was also mentioned as one of those species. There are a lot of species which have been introduced and they have become as good as native species in those areas. But what happens here is there's something called enemy release hypothesis, where the success of a establishment of a species is governed by, say, if you leave your pathogens, your competitors, basically, when you move to a new habitat, because it gives you a kind of an advantage. And we wanted to look into how does that translate into the number of parasites. There's a lot of work being done on plant pathogens as well. So if you move the plants to a new location, they kind of thrive, some of the species, because they are not experiencing same kind of diseases which they face. And here what we found that the common manas, this is the native range of common manas. These are all the locations where this bird has been introduced. Why do people introduce birds? I don't know, don't ask me. They just release birds and they introduce. We found that a lot of plasmodium parasites, what they carry here in Australia, they had a very low, which is in the introduced range versus in the native region. But it kind of flips into for other host-specific parasite, which is more or less stays equal. So it changes with the kind of parasites, with what you see here. And finally, one more thing I want to bring in here is that a lot of species, like there could be a parasite sexual selection, which might be playing a role for the parasite prevalence. So if you have a male and female, depending on who is sitting on the nest for longer, might be exposed to more vector species, might be having a higher infection rate. Same for the body condition here. So here we found that, if you remember that Umesh was mentioning that you can do the wing length and the length of the tarsus. That actually allows you to calculate the body condition for a bird species. And we found that the uninfected birds had a slightly better body condition than the infected birds. This is to do with the bird malaria. So there are lots of ways when you, I mean, if you have a bird in hand or any animal, you can do lots of things. But if you don't, then looking for non-invasive way, I think is the only way to do it. Now, this is the last slide I have, and I think this is a very interesting thing for me. So in England, if you know that if you go to England, everybody have bird feeders in their gardens. It's like part of their culture. They just they feed birds, they feed a lot of birds. There are Royal Society of Protection Bird, they sell a lot of bird food. And people like birds, it's like a normal thing to have. But as a result of this bird feeding, so some of the species starting contracting one of the protozoan parasite called trichomoniasis, which led to the decline of this bird. So one more other thing is to see a decline is happening, you need to have a lot of long-term data, and they do monitor their birds very well. They have a very good bird breeding atlases or distribution atlases, which allows them to see that how one of the species of this chaffinch, how this population started kind of going down here, and for another one like this. And then they kind of mapped it that what kind of areas this was happening more. So they found that in the peri-domestic areas where the feeders are more, they are actually driving this infection more. And being a social species, because all these birds, they come and they utilize the same place for feeding, they are getting this infection back and forth. And these bird feeder gets other species as well, but these species are the only ones which are prone to these infections. So that's something I want to highlight here. So being a protozoan parasite, probably it was not infecting or problem for other species, but for these two species of chaffinches, which it was a huge problem, there's a huge decline. They are kind of become in their red list, if I call them, national red list. So some of these questions, what we are looking at in the disease ecology are very fundamental. It's just not about going to a forest, doing the work. It could be your neighborhood looking for mosquitoes habitat and how infections might be driving in the population, and number of ways one can try and understand. I'm going to stop here now. If you have questions, we can take it now, or how you guys feel. Everybody looks tired right now.
There was this one study, I'm not sure if it's part of this one, but they mentioned how the bird feeders.
Sorry.
Thank you. They mentioned how the bird feeders tend to get a lot of fungus around the openings where the birds usually feed on, and they're all getting this fungal infection around their beaks. So is it part of the study?
There are lots of ways how they have done. I mean, also, you're right. I mean, and also one of the things they kind of told they do garden watches and things like that, and the whole community, like all the 70 million people get involved in that. There was a mass movement, please clean your feeders. That's the fundamental thing you can do. Change the food and all of that kind of thing. Yeah. So while you all are thinking, I just want to put this here. I think Robin mentioned the book in the morning, Song of Dodo. If you are really interested in understanding how the spillover and other things, this is a lovely book to read. It's very, very nice. I have read it back to back. It talks about all the Hendra virus, how they discover. It's like a detective work, figuring it out. Goes through different kind of scenarios for different diseases. So please make it your bedtime reading. You'll love it. Yeah.
I just have one question. I mean, not a question. I just wanted to hear your thoughts about supplementary feeding. There's also the school of thought that supplementary feeding, especially during winters, is beneficial to wild birds. And that kind of adds up, like in whatever nutrition that they're missing out on. So I just wanted to hear your thoughts on that.
No, definitely. I mean, even though this decline and whatever happened, it happened in certain European other countries as well. What they were not saying that you don't feed. They just said make the hygiene as the part of it. Don't just leave the food out there. Make sure it is clean, basically. Yeah, they never. But yeah, a lot of winters, when it gets too cold or something, yeah, I think that becomes a very crunch period for a lot of species that time. Yeah. I like birds. Don't ask me. I'll feed them.
Yeah. Ma'am, just a doubt. Want to correlated situations happening recently in Kerala. So you were telling about the situation when a virus happens, like get infected, it won't be prevalent in a particular year. So the cases won't be there in a particular year. So the next year it can be prevalent. So it is because that you said it can freeze the virus. That's why that was not affected in that year, and it's getting unfrozen. It will be affected.
In Kerala?
No. That was the question.
When I say in Mongolia.
Yeah. So now the recent Kerala and Kuttanad. So this is happening. The H5N1 has been outbreak. So that time there was no migration. Migratory season was not there. All of a sudden,
Poultry.
Poultry. It was all affected in ducks. Only it was seen in ducks, domesticated ducks.
Do you know that a lot of these domestic wild ducks and also even poultry, generally birds in general, especially these, as I call them, the ducks and geese, they are naturally reservoir of lots of viruses. I mean, especially influenza viruses, they carry all of them. They're just born, evolutionarily evolved with them. H5N1 happens to be a very pathogenic one, which also kills them. Any H5 and H7, by the way, hematoglutinin, which makes it the combination, whichever combination it happens. And that usually happens when they come in contact with some other strain in a very crowded situation. That's when this kind of antigenic shifts is happening. H1N1, swine flu, which also happens in the same situation where you have different species coming in contact. So it becomes like a flu factory, you know. So if you have a subclinical infection going on, so say for example some of the migrant species, even while infected with some of the H5N1, they managed to make that long journey. They might die towards the end of it, but they survived enough to be on that spot to shed the virus, and by the time the outbreak kind of thing, I'm not saying only one individual, maybe few coming together. So these things happen. I mean, it depends all on the individual how much they can. Some maybe not be showing you any symptom, but they are happily. Somebody might be COVID positive in this room right now, you don't know. I got infection 10 days ago in a meeting. So yeah, these things happen, that's what I'm saying, yeah. Any other question? T?